Test, test, test. Wow. Seven people. We're down a few today. Well, as my dear mother used to say, it's not quantity, it's quality. So with that in mind, we will go ahead and start our lecture for the day. We have, I guess we have 1 minute to go. Maybe we'll wait. Jacob, good morning.
Good morning.
And here comes Jessica. Morning, Jessica. And now it's 12:20. We'll go ahead and begin. Lord, thank you for this cold day. We thank you for the sunshine. We thank you for a season to come together and give thanks. We thank you for the chance to learn, even though it's under not very ideal conditions. Give us this love of learning. Give us this love of knowing how our human bodies work so that we can be better stewards of it and we can help others perchance who aren't as good a steward or perhaps have fallen on rough times, need a hand up, Lord. Some of us want to be healers. Some of us just want to be people who read the user's manual a bit better. So, Lord, now, perk us up. Give us a chance to have more energy, perhaps. Help these students be more participatory today than they might normally be online. Bless this time now in Jesus' name. Amen. Okay, so last time we finished with a discussion of the structure from a macro perspective of the small intestine. Let's take a little bit of a look at the micro anatomy of that, if you will. So not only is the intestine itself folded into these plicae, and then on those plicae you have villi, but if you look at the individual villi in each of these cells, you actually have at the apical surface, so that's the apical surface, this would be the basolateral surface. The apical surface is also folded into so-called microvilli. This further increases the surface area, the opportunities for absorption, and the absorptive capacity of these cells. Now, let's blow this up even more. So I'm going to take this little piece here, and we're going to blow this up like this. And here's what we're going to see. We're going to see a piece of the membrane, essentially, and I told this to another class. I should probably tell it to you. The one class I almost failed in middle school was art, and you can see why. All right, now attached to this, you have these enzymes that stick outside the membrane here. and, what have you. These enzymes essentially are in intimate contact with the chyme. So the chyme essentially is going to come in contact with these enzymes. These enzymes will break down dipeptides, tripeptides, disaccharides, things like that. And these are called brush border enzymes. And that's based upon this looking a little bit like a paintbrush. So these brush border enzymes take larger macromolecules that the cell can't absorb, breaks them into monomers that now the cell can immediately absorb. And this makes digestion much, much, much more efficient. Brush border enzymes at the microvillar surface that is found at the apical surfaces of these enterocytes. And of course, over here, we have also a basement membrane down here. Like all good epithelia, this sits on a basement membrane. But then from that basement membrane, these cells are going to take these monomers, they're going to send them straight into the bloodstream. Like that. So essentially we go from these macromolecules to monomers to enterocyte to blood and straight into the circulation. Very, very efficient system for absorption. So if we look at some of the brush border enzymes, we have things that break down disaccharides, sucrase, maltase, lactase. Sucrase degrades sucrose to fructose and glucose, two monomers. They'll be absorbed. Maltase, which degrades maltose, which is a disaccharide with two glucose molecules hooked together, to glucose, which is absorbed. Then lactase, which breaks down lactose, which is found in milk, to galactose and glucose, which are absorbed. And some people's genetic heritage will cause their lactase levels to wane as they get older. And they will suffer from a condition known as lactose intolerance. If your ancestors were cow herders and people who drank the milk of cows, then your lactase enzyme doesn't wane. And it's due to a single base change in the promoter of the lactase gene that causes it to persist into adulthood. So amongst the Bantu tribes of Africa, those people herded cows and drank the milk, and also amongst Northern Europeans, Scandinavians, Dutch, Northern Germans, and some of the French, Northern French, you have this capacity to drink milk into adulthood. Unfortunately, my Eastern European heritage has no such advantage. Other brush border enzymes include aminopeptidases that degrade peptides from the front end, amino acid by amino acid, to produce free amino acid, di and tripeptides. Enterokinase, which is an activator of zymogens made by the pancreas, and then phosphatases and things like that to take phosphate groups off molecules that have phosphates on them. Casein, for example, which comes from milk, is heavily phosphorylated. Alkaline phosphatase produces phosphate that can then be brought into the cell, and then other types of ATPases for bringing in calcium, magnesium, and other types of ions as well. These are all brush border enzymes. If we look a bit at the not just a smooth muscle, but the nervous system that innervates the smooth muscle. So SI here is small intestine. It does not mean Sports Illustrated. So the small intestine has two major contractions. There are peristaltic contractions. These occur because the pressure at the caloric end is greater than the pressure at the more distal end. And then segmentation, which is contraction of that circular smooth muscle in order to move the food bolus along. There are two main types of muscle contractions in the smooth muscle of the GI tract. They're so-called tonic contractions. Tonic contractions. These are continuous. And then you have phasic contractions. In phasic contractions, the smooth muscle will contract and relax in waves. The contraction and relaxation in waves tends to produce peristalsis, and then the more powerful phasic contractions are responsible for segmentation. The cells of the myenteric plexus have certain interstitial cells that act like pacemakers. These are called interstitial cells of Cajal. Interstitial cells of Cajal. You see them there, or ICC. Interstitial cells of Cajal. These cells can generate rhythmic depolarizations and repolarizations of the cell membrane. These are called slow wave potentials. They can do so without any external stimulation from the neural or the endocrine system. And you can see the electrophysiology of these slow waves. They're very regular. You can kind of sort of set your watch by them. These can be influenced by input from the sympathetic and parasympathetic nervous system. But the intrinsic capacity, that is not something that is caused by the sympathetic or parasympathetic nervous system. This is intrinsic to the interstitial cells of Cajal. So they do this by means of a slow sodium channel, which is in the cell membrane of the interstitial cells of Cajal. These will cause an influx of sodium, and then repolarization is caused by the opening of potassium channels, and that causes an efflux of potassium. The Cajal cells, as you can see here, are connected to smooth muscle cells via gap junctions. So the slow wave potential will travel to the smooth muscle cells. That will trigger voltage-gated calcium channels to open, causing depolarization of the smooth muscle cells. Slow wave potentials will cause relatively weak depolarizations. They don't reach the threshold potential needed to cause peristaltic contractions, but they are strong enough to cause the tonic contraction, which will maintain the tone of the gastrointestinal tract. Action potentials from the extrinsic nervous system will cause depolarization of the smooth muscle cells by opening a different set of voltage-gated calcium channels. When these extrinsically triggered depolarizations are combined with the slow wave depolarizations, now they will reach the threshold to trigger peristaltic contractions. And notice that the autonomic nerves have these varicosities as the action potential passes down these nerves. They will release neurotransmitters on the pass, en passant, and that will bring these depolarizations actually up to threshold, causing peristalsis. So the tonal contractions, those are intrinsic. Phasic contractions necessary for peristalsis, those require the tonal plus the extrinsic input from the autonomic nervous system. And because the smooth muscles tend to be connected with each other via gap junctions, you'll get spreading from one cell to another. It's worth saying here, different sections of the gastrointestinal tract have their own unique slow wave frequency. So for example, the stomach has the lowest rate of 3 slow waves per minute, but the duodenum has the highest rate at 12 slow waves per minute. So the speed is going to differ from one part of the GI tract to another.
Dr. Bradwich, can I ask a quick question?
No questions. You may not, of course you can. Go ahead.
Are these contractions happening even when, like, say we haven't eaten for a long period of time and there's very little in our stomachs? Are these contractions still happening then at all times, or is it only when there's stuff to contract?
Yes, they're happening. They are happening, but when you eat, you activate the parasympathetic nervous system, and eating, that activation of the parasympathetic nervous system by eating will cause increased contraction. Now, also, you may have noticed this, if you exercise, that tends to shut down the GI tract, but then when you're done exercising, because your sympathetic nervous system is now downregulated, your parasympathetic nervous system sometimes is upregulated. Therefore, sometimes after you work out, you may discover you really have to go, and that's due to renewed, stronger activity of the parasympathetic nervous system. So it's not just eating, it's other things that can activate the parasympathetic nervous system, and that's why exercise will tend to help you relieve constipation sometimes, because of that upregulation of the parasympathetic nervous system after exercise that occurs. Does that answer your question?
Yes, thank you.
Drugs can also activate it too. Cholinergic drugs. For example, if someone's completely plugged up, there's this drug you give them called bethanechol. Bethanechol is a major-league cholinergic, and the bowels of heaven will be opened when you take bethanechol. Trust me. So yeah, that's an example of drugs actually doing this. And some cholinergic drugs, for example, drugs that are given to Alzheimer's patients are cholinergics. Those will tend to cause diarrhea because of that. And I just said all that. So the contractions are modified by the autonomic nervous system. Acetylcholine from the parasympathetic nervous system will increase the amplitude, duration of the slow waves. Norepinephrine, epinephrine from the sympathetic nervous system will tend to decrease the activity from the intestines. And I'll just keep that up. 3, 2, 4, 1, and then there. We'll move on. Now, for peristalsis, you have this very interesting regulation of peristalsis. So here is a food bolus. You have mechanoreceptors that detect the distension of the intestine. These will then connect to interneurons. The interneurons are in the blue. The interneurons here upstream, as you can see here. So these upstream these are going to send acetylcholine up here. Acetylcholine is going to cause these to release substance P and acetylcholine, and these will cause relaxation. Oh, wait, sorry, which direction? Oh, no, sorry. These will tend to cause contraction. Yeah, your food bolus is moving in this direction like this. So this will cause contraction, whereas this one is going to go in this direction, and this will tend to activate neurons that release nitric oxide, vasoactive intestinal peptide, and ATP. These will tend to cause relaxation. So these will tend to be inhibitory when it comes to smooth muscle, whereas substance P, acetylcholine, will tend to be excitatory. These cause hyperpolarization or relaxation. So you get constriction here, relaxation here, that propels the food bolus in this direction. So your sensory neurons send different directions depending upon their distension. Upstream, they will tend to cause contraction. Downstream, they will tend to cause relaxation. Food bolus moves, new set of mechanoreceptors upstream here will cause contraction. Downstream there will cause relaxation. You're always causing contraction upstream, relaxation downstream. You're propelling the food bolus, therefore, in one direction. So acetylcholine, substance P, stimulate smooth muscle. Stimulate smooth muscle. Whereas nitric oxide, vasoactive intestinal peptide, ATP, stimulate relaxation. And now the food bolus will move down. That's how come the food bolus moves in one direction only. Okay, so the absorption is something that occurs mainly in the small intestine. We'll talk about that when we talk about each of the different types of nutrients that get absorbed. Let's go now to the large intestine. So the colon or large intestine extends from the ileocecal valve through the ascending, through the transverse, through the descending, sigmoid, colon, rectum, and then to the anus. And then the chyme passes from the ileum to the cecum. And then it passes through these parts of the colon. And then the material that will pass through the anal canal is known, of course, as fecal material. So here is the overall structure of the colon. Here's the ileocecal valve. Here's the ileum. Remember, the ileum is about 2/5 of the small intestine. Jejunum is about 3/5. The duodenum is pretty small. Then you have the cecum. Here, this is an expansion of the colon here. You have an extension of this called the vermiform appendix. This is loaded with lymphoid tissue. If you talk to surgeons, the size of the appendix varies greatly from person to person. As you go from childhood to adulthood, this tends to fibrose over. If you don't have it fibrose over completely, you can have material that gets caught in here, and that can cause ischemia of this tissue, resulting in gangrene. That's, of course, appendicitis. That requires surgical intervention. Some people, the appendix is very short. Others, the appendix is really, really long. Varies from person to person. Here's the ascending colon. The ascending colon is wider than the descending colon. So if you have a, anyway. So you can see there's this structure along here. This is called the tinea. These have these little extensions called epiploic appendages here. These are probably lymphoid tissue. And then these bulges, these are called haustra. And then you have this is the hepatic fissure over here. There's a flexure, hepatic flexure. Then this is a splenic flexure over here. Then the sigmoid colon, rectum, and then anal canal over here. This is held in place by the mesentery. This is filled with lymph nodes and also a very rich blood supply that's supplied by the superior and inferior mesenteric arteries. You don't have villi in the large intestine. Instead, the mucosa is organized into these beautiful columns, and these columns are called intestinal crypts. Intestinal crypts, that's what CR stands for there. You have lymph nodes, vessels, things like that. Here's your muscularis mucosa right here. Here's your crypts. There is an extensive series of lymph nodes. So LN, that's the lymph node there. This is called the gastric associated lymphoid tissue. Gastric associated lymphoid tissue. Here's your column. You can see it almost forms a little pit. And these have, you can see right there. Let's see, can I blow this up? Can you see right there? Those are goblet cells. Can you see those? There's one, there's one, there's one, there's one, there's one. Hope you can see that. And then there's lots of mucus in here. Material essentially will come in here and be subject to further absorption and what have you. Also, these lymph nodes are constantly surveying material that comes in here, and the gastric associated lymphoid tissue is more highly developed in the large intestine than in the small intestine. And here's a really beautiful cross section that shows you essentially these beautiful lymph nodes here. Lots of these here. So there's one, there's one, there's one, there's one, there's one. So this tissue, very heavily associated with immune surveillance in the intestine. So it gets absorbed in the large intestine. You have, first of all, several fat-soluble vitamins will be absorbed. So vitamin K is absorbed. Vitamin K is produced by bacteria in the GI tract. If you get it from your diet, it's also absorbed there. Some B vitamins like B12 also gets absorbed there. The remaining water will be absorbed in the large intestine. as will some of the other things that are, as we'll see, bile salts, they get reabsorbed in the large intestine as well. And then also iron reabsorption is important also in the large intestine also. So the large intestine will absorb some 90% of the volume that remains in the chyme. Therefore, fecal material tends to be relatively dry. That limits water loss. It's of course subject to level of hydration. It's also subject to how much fiber, indigestible fiber, is in the chyme as well. So water absorption, you don't directly absorb water. You absorb ions, and then water comes along for the ride. So you don't have active transport of water. You take up salts, and then water follows. Under some circumstances, water and salts can be actually excreted. So for example, Vibrio cholera, there's an enterotoxin made by Vibrio cholera that stimulates active transport of sodium and chloride. Water follows, and you can lose enormous amounts of water in a kind of rice stool diarrhea. And you basically shrivel up like a prune and die. It's an awful, awful way to die. And fortunately, there's a rehydration therapy that was interestingly developed by a nurse that can help with that. That's why some of you who went on trips to some parts of the world, you had to get a cholera vaccination. Celiac sprue is caused by a significant type 2 allergy to gluten. So gluten ends up getting across the epithelial border. It's then presented to the immune response, to the immune system, excuse me. You then have a robust immune response against it. You have antibodies secreted against the gluten, and that inflammation causes secretion of electrolytes into the bowel, and that causes celiac sprue. Lactose intolerance, due to an inability to secrete lactose, so the bacteria in your GI tract break it down. That causes the production of organic acids. That will cause secretion of lots of salt and water and can cause diarrhea and lots of unpleasant bloating and stuff like that. And these conditions that cause water loss from the GI tract, they're collectively known as diarrhea. Defecation is the result of increased rectal pressure. You have two sets of muscles. You have a so-called internal anal sphincter and an external anal sphincter. You also have a set of so-called hemorrhoid pads. So the defecation reflex begins with distension of the rectal walls, which is caused by movement of material into the rectal canal. These will cause, let's go to the, come on, you. Computers are great when they work. Okay, so and you have so this goes essentially use your imagination, pretend this really ugly thing is your spinal cord. So you have essentially sensory neurons here. They connect here. Should have done that in the same color, but yeah, too late now. All right, so you have connections. So you have the sensory neurons are going to come in. They will then connect to the motor neurons that are going to connect to the muscles, essentially here. And these also are going to connect. And you'll have also connections from higher order neurons up in the so brain. Handwriting is really starting to, that says brain. I know it looks ugly. Okay, so the sensory neurons detect distension. Sensory neurons here as well. So these parasympathetic motor fibers will stimulate contraction of the rectal walls and relaxation of the external and internal anal sphincter. So these structures here essentially are under control of the sympathetic nervous system. So the autonomic nervous system here. If it is convenient, then to defecate, voluntary signals will cause relaxation of the external sphincter, which is out here. That voluntary connections here, if it's not convenient to defecate, then it can be delayed by essentially conscious cortical controls, which allow voluntary constriction of the external anal sphincter here. So these are under the control. The autonomic system here, that's controlled due to sensory input from mechanoreceptors in the wall of the anus. there is a portion of the anal canal and this is called either the dentate line, above which you don't have a lot of pain receptors. That's why if you have surgery, the anesthesia only needs to be given to this area, doesn't need to be given to this area. This area is derived from ectoderm. During development, this is derived from endoderm. So the pain reception here is very different than the pain reception down here. Okay. There are also in here some vascular cushions. There's 3 of them. I've drawn 4 of them, sorry. 1, let's say this one, 2, 3. These are called hemorrhoid pads. They fill with blood, and when you cough, these are intended essentially to prevent you from soiling yourself when you cough. If these become inflamed or damaged, you can get vascular backup. That can cause the veins in this area to swell up. That's what causes these things we call hemorrhoids. Or you can get things in here, and that's sometimes what we call piles. And those can bleed sometimes. That's what causes a so-called bleeding head. So histologically, the rectum has kind of 2 parts. There's an upper bit that resembles the colon, and then the lower part that does not. And above the anal valves, the mucosa is very columnar, has goblet cells, has crypts, so on and so forth. And below that, you start to get more of a stratified squamous epithelium that can handle shear forces much better. And you can see this here, this transition from the upper to the lower anal canal. See how this now starts to get stratified, whereas here this looks more like upper part of the large intestine, excuse me. BB, that stands for blood vessels. Okay. All right. Let's talk about the liver. So the liver lies just below the diaphragm in the right upper quadrant of the abdominal cavity. It's the largest internal organ you have. It does a wide variety of things. It manages the body's metabolism, does detoxification, makes bile, makes most of the blood proteins. The surface of the liver is covered by a serous membrane called the visceral peritoneum. And the peripheral visceral peritoneum folds over on itself, and it suspends the liver from the abdominal wall and the diaphragm. the liver come on, computer. So this is the backside of the liver. The liver has a series of these peritoneal folds that hold it in place. Those are called ligaments. And one of those ligaments, the falciform ligaments, you can see it here, breaks the liver into the left and the right lobe. You can see the right lobe is much bigger than the left lobe. And if you view the liver from below, you have this two additional lobes in addition to left and right lobe. You have the caudate, oh, stop it, you. You have the caudate lobe here and the quadrate lobe back here, but the thing I want you to see is what separates these two lobes. This triad of vessels and ducts, this is called the porta hepatis. Porta hepatis. And that means gate to the liver. Gate to the liver. So it contains the hepatic artery, the hepatic portal vein, and the common hepatic duct. The common hepatic duct. Common hepatic duct in green there drains bile from the liver into the gallbladder. The hepatic artery here in, looks to me like orange, delivers oxygen-rich arterial blood from the heart to the liver. And then your hepatic portal vein delivers nutrient-rich blood, venous blood, from the GI tract. And also blood from the spleen and the pancreas goes to the liver as well. So, lesson learned here is this. After materials are absorbed by the GI tract into the blood, what's our first stop? Answer, the liver. The liver. Okay, a little quick anatomy lesson there on the liver. Now, if we chop the liver into pieces and look at what the little pieces look like, we see the liver is broken into these functional units called hepatic lobules. Each hepatic lobule looks like a little tiny hexagon. And if you ever look at slices of chew, if you see the hexagonal organization, that's a dead giveaway, it's liver. So let's take a look at these little hexagonal structures. These hexagonal structures are called hepatic plates. Hepatic plates. And together they form a hepatic lobule. Hepatic lobule. So the cells that constitute the hepatic plates are called hepatocytes. Hepatocytes. Then you have so-called portal triads. You can see the portal triads over here. And here's our portal triad right there. So you have a branch of the hepatic artery, a branch of the portal vein, and one or two small bile ducts that will empty into the larger bile duct. Now, the branches of the hepatic artery and the portal vein are going to drain into this very porous blood vessel that's called a sinusoid, hepatic sinusoid. You see them labeled here, the sinusoid. You'll notice here how these will kind of drain here like that. they will then empty into the so-called central vein. So the blood here goes here this way. Arterial, venule, into the central vein. From the central vein, the blood flows into the hepatic vein, and then it will drain from there into the inferior vena cava. Okay, back to these sinusoids. Now, oxygen and nutrients will get through the pores of these hepatic sinusoids and enter the underlying hepatocytes. So the blood will be, the hepatocytes will be exposed to this blood, and they're exposed to this blood continuously. Continuously. That's one of the things I want you to see about this. They will also pick up and detoxify harmful substances like drugs or alcohol or something like that. They'll also help maintain a normal blood glucose level. After eating a meal, they'll take the glucose and convert it into glycogen. That's called glycogenolysis. And if the blood glucose levels are low, they'll break down the glycogen, secrete it into the bloodstream. That's called glycogenolysis. The hepatocytes will store certain vitamins, fat-soluble vitamins like A, vitamin D, E, K, and vitamin B12. They'll also store some minerals like iron and copper. And they'll synthesize important blood proteins like albumin and clotting factors. They'll break down fats. They'll also process amino acids from proteins. They'll take the amino group off those amino acids. Then they'll use the amino groups to make urea. They'll also synthesize from fatty acids and cholesterol very low density lipoproteins or high density lipoproteins. Also, another thing about the liver, you could donate 90% of your liver and it would grow back. This is a tissue with tremendous regenerative capacity. All right, so I hope I hammered that home. Food absorbed in the small intestine is first delivered to the liver. So let's underline that so everybody knows that's really important. Okay, then we talked about what the blood supply does. The sinusoids drain into the central vein, which then receive blood from the hepatic artery, goes then back to the vena, so on and so forth. So the hepatic plates are the functional structure of the liver. Now, one other thing about hepatocytes. Hepatocytes make bile. While blood is flowing in this direction, bile is flowing this direction. So bile is going away from the hepatocytes. Blood is flowing toward the hepatocytes. Very important to remember that. And this essentially shows you the flow of those materials. Artery, vein, yes, arterial venous blood mix, but that's for the sake of the hepatocytes. And then hepatocytes also make bile from cholesterol. They then send that to the bile ductule. and the bile ductules will fuse to form the right and left hepatic ducts, which will merge to form the common hepatic duct, which then leads to the cystic duct, which brings the bile to the gallbladder. Okay, and we have a particular type of recycling of certain compounds back and forth to the liver from the intestine. They're absorbed back again via the hepatic portal vein, back then into the bile, down into the GI tract. They're reabsorbed, and this is called the enterohepatic circulatory system, enterohepatic circulation. And this summarizes some of the functions of the liver that I have tried to summarize for you. Okay, questions about the liver before we talk a bit about more cool stuff the liver does? No? Okay, let's then talk about bilirubin. So bile contains bile salts, water, and bilirubin. What is bilirubin? Bilirubin is a breakdown product of hemoglobin. So the cells, red blood cells, after they have served their purpose, the red blood cell will be taken up by these stationary macrophages that are found in bone marrow, liver, and spleen, mainly in the spleen. So reticular cells in the spleen. In the liver, you have what's called a Kupffer cell, K-U-P-P-F-E-R, Kupffer cells, and then just stationary macrophages in the bone marrow. They take the red blood cell, they break it down, they degrade the globin to amino acids. Amino acids get recycled or digested and used for energy. But the heme is very insoluble and has to be handled differently. So the iron is taken out, recycled. The heme is then converted into biliverdin, which is then converted into bilirubin. Bilirubin. And then bilirubin, it hitches a ride on albumin and gets carried to the liver. Bilirubin is not very soluble, so getting rid of it takes some finagling. Okay, so your liver is going to take some of that bilirubin and conjugate it to glucuronic acid. It'll form conjugated bilirubin. This is charged. That's what then gets solubilized in water and gets put into bile. And in the intestine, about 30 to 50% of that bilirubin, okay, first of all, the conjugated bilirubin in the intestine gets taken back up via the MRP2 transporter, and it enters the hepatic vein and goes back to the liver. And it'll be recycled, or it'll get filtered by the kidneys because it's now soluble, so the kidneys can now excrete it. But intestinal bacteria, members of the genus Bacteroides, convert conjugated bilirubin into urobilinogen. Urobilinogen. Now, urobilinogen can be oxidized to produce a compound called urobilin. Urobilin. And then the rest of is converted to stercobilin. And stercobilin is brown, and that's what's responsible for the brown color in fecal material is stercobilin. Sorry, stercobilin. And that is usually the result of bacterial activity. Now, what if your liver is having trouble processing bilirubin? The liver, if it now gets overloaded and it can't put enough of this bilirubin into the conjugated form, that bilirubin now is going to spill over into the bloodstream, and it starts to get deposited in tissues. And one tissue that really has a high affinity for free bilirubin is the conjunctiva of the eyes. And then it gets deposited in the skin, and bilirubin is yellow. That produces this condition called jaundice. Jaundice is indicative of liver failure. Free bilirubin will also get excreted into the urine, and there's a nice little trick you can use that will tell you if there's free bilirubin in the urine. Take the urine sample, make sure the lid is on it nice and tight. There'll be some foam in it, and shake it. And if the foam turns yellow, there's bilirubin in the urine. Otherwise, the urine should still stay white. That's a little lab trick that's still used today. There are easy ways to measure bilirubin, but if you just want a quick, is there bilirubin here? That'll tell you right away if there's bilirubin. All right, so this gives you the pathway essentially from biliverdin to bilirubin. Bilirubin then hitches a ride in albumin to the liver, then it's conjugated. By UDP glucuronyl transferase, or UGT1, and it gets excreted. Some of it gets reabsorbed, some of it stays in the GI tract and is subject to modification by bacteria. And then cholesterol is used to make bile salts. They're conjugated to either glycine or taurine. So the cholesterol is converted either to cholic acid or chenodeoxycholic acid. Then you conjugate them to glycine or taurine to make glycocholic acid or taurocholic acid or glycocenodeoxycholic acid or taurochenodeoxycholic acid. And then you make the salt version of those, and they are then excreted into the small intestine as bile. And those get reabsorbed, some of them, others don't, and are excreted out in the fecal material. Okay, it's a good spot to stop. We talked about some of this already. We'll get through the liver and then talk about other features of the GI tract, and that should do it. You guys all please have a terrific Thanksgiving. Please remember to be thankful, especially for your families, and I will see you guys all on Monday. Blessings, everybody.
I have a couple questions about the quiz.
Yeah.
I think from talking to other people too that one of the answers was wrong.
I fixed that.
Okay. And then there was another one that seemed like it had two correct answers. It was the one regarding the diver.
Yeah, but I asked you to pick the best answer. There's one answer that's better than the other. Pick the best answer, Meredith.
Okay. I guess I just didn't really realize that one was better than the other.
Yeah, pick the best answer.
Okay.
You pick the best answer, The MCAT probably was that way at times. It looks like there's 2 right answers. We just have to remember, pick the best answer.
Okay.
Okay?
Yep.
All
Does
right.
that the one that was marked wrong, does that get corrected on our?
Yeah. Blackboard has this wonderful component. If you change an answer, it updates all the scores.
Okay, cool. And then
And
I also had a
did that.
question.
I did that just before class today.
Sweet. Moving forward, after we finish the digestive system, are we continuing with the order that's on Blackboard, or are we going to jump around?
Well, I'm
certain-
probably going to finish digestive system via a podcast. Then we'll do the kidney. Kidney's quite important. And then I'm guessing we'll probably get to start reproductive. You may just have to rely on the videos I have online for the rest of the reproductive system. We'll probably skip metabolism.
Okay. Cool. Thank you.
Okay? All right. Monday, gang. Happy Thanksgiving.